Welcome, curious minds, to the deep dive. I'm your host, Alex, and today we're embarking on a really fascinating exploration. Okay, let's unpack this. Today's deep dive is into PT-141, also known scientifically as bremelanotide. Now, this is a research peptide that has, truly sparked interest in neuroscience and specifically arousal research. What makes PT-141 so compelling, kind of groundbreaking in this whole field is its action. It's really distinct. See, unlike a lot of the traditional approaches that focus mainly on, say, circulation, just getting blood flow going match, PT-141 has been studied for how it seems to work directly within the brain's own central arousal pathways. So think more top-down influence, maybe. Right, so our mission today is basically to get you well-informed on this pretty complex topic, give you that shortcut. We'll look at its origins, which are quite unconventional, actually. We'll dig into the mechanisms, the surprising twists and turns in the research, and hopefully uncover some aha moments about what it means for human biology. Get ready.
To guide us through all this intricate stuff, I am absolutely thrilled to be joined by our expert guest today, a neuroscientist specializing in neuropeptides and behavior. Just brilliant at connecting the dots on complex topics. Welcome.
Thank you so much for having me. I'm genuinely excited to dig into this. You've really hit the nail on the head already. What's truly captivating about PT-141 is how it challenges our usual ways of thinking. For so long, when we thought about arousal, particularly clinically, the focus tended to default to peripheral mechanisms, right? What's happening physically in the vascular system. But PT-141 kind of forces us to look deeper, inward, towards the central brain mechanisms. And that raises this really crucial question. What if desire, where it actually starts, isn't just about blood flow? What if it's really about brain chemistry, about the neural networks that kind of orchestrate motivation and reward? It suggests something more fundamental.
That's a really profound way to frame it. Like maybe we've been focused on the symptoms, not the source.
Possibly, yes. Or at least not the whole picture.
It definitely makes you wonder. Okay, so let's start from the basics then. What exactly is PT-141? Where does it fit in?
Certainly. Okay, so at its most basic level, PT-141, or bremelanotide, it's a synthetic peptide. Now, synthetic just means it's made in a lab, but importantly, it's derived from a natural family of hormones. These are called the melanocortins, and they're actually biologically very significant, quite ancient. Melanocortins are involved in a whole bunch of different processes. A lot of people might know them for pigmentation, skin, hair color, that sort of thing. But their job goes way beyond that. They're key players in regulating appetite, energy balance, metabolism, even things like inflammation and immune responses. So they're pretty fundamental to how we function overall.
Right, so PT-141 is basically an engineered version, an analog of one of these natural melanocortins, specifically alpha MSH. But the key thing, like we mentioned, the real differentiator is its central action, meaning its main effects are studied within the brain and central nervous system, not directly on peripheral tissues or, the vascular system like some other approaches.
Okay, so it's less about adjusting the local plumbing, so to speak.
And more about influencing the brain's command center, tapping into something more fundamental about our drives.
Precisely. It's about influencing the neural blueprint for desire, which actually leads us nicely into its rather unexpected origin story.
Ah, yes. That's the part I heard about, that it started in tanning research. Sounds like pure serendipity.
It really does. A classic case of science taking a detour.
Tell us about that. How did they go from trying to get a tan to studying desire? What was that aha moment?
Well, you've got it right. The initial research had nothing to do with sexual desire. It was all about pigmentation. Scientists were looking at different synthetic versions, analogs of alpha MSH, that melanocortin we just talked about. The goal was pretty straightforward. Develop something that could safely stimulate skin tanning, like a sunless tan from the inside out. So picture these early clinical trials, phase 1, phase 2. You've got healthy volunteers. They're getting this experimental tanning peptide. And then something completely unexpected starts popping up in the feedback. A number of participants, both men and women, started reporting, increases in sexual desire, sometimes spontaneous erections, heightened arousal.
Wow, really? Just out of the blue?
Pretty much. Initially, maybe it was brushed off, an anomaly, but the reports kept coming. It became too consistent to just ignore. That was the critical aha moment,
Wow.
This totally unintended side effect. It basically forced the researchers to pivot, to shift focus from skin tanning to this the central effect on arousal they'd stumbled upon.
That's incredible.
It really is. So then the challenge became, okay, how do we refine this peptide? How do we separate out the tanning effect from this new interesting effect on desire? It involved a lot of careful molecular engineering tweaking the structure. The aim was to make it bind more strongly to the specific melanocortin receptor thought to be involved in sexual function. That's the MC4R. We'll talk more about the mean. While at the same time making it less active at the receptor primarily linked to tanning, the MC1R.
So fine-tuning it, essentially.
Exactly. Fine-tuning the key for a specific lock. It's really a great example of scientific discovery. You start looking for one thing, find something totally different, and end up enriching our understanding of, well, human biology in a whole new way.
That is just fascinating. The journey from A to B wasn't straight at all.
Not in the slightest.
Okay, so they refine it to target these desire pathways, specifically MC4R. This is where it gets really interesting for me. How does it actually work? Inside the brain. What's the mechanism behind this central switch you mentioned?
Right. This is really the core of PT-141's novelty. It's all about engaging the brain's own arousal pathways. So after it's administered, PT-141 needs to cross the blood-brain barrier, which is a feat in itself, and then it interacts with these specific receptors, primarily the melanocortin 4 receptor, MC4R. And these MC4Rs are found in high concentrations deep within the brain, especially in the hypothalamus.
The hypothalamus. I know that's a really key area for basic drives, right?
Absolutely critical. It's small, but it's like a master control center. It regulates hunger, thirst, sleep, body temp, and crucially for us, motivation, reward, and behavioral drives, including sexual function. It integrates signals from all over the brain and body.
Okay, so PT-141 hits the MC4R in the hypothalamus. Then what happens?
Well, the proposed mechanism, what the research suggests, is that by activating these MC4R circuits in the hypothalamus and related areas, like the limbic system, which handles emotion, PT-141 seems to modulate the release of key neurotransmitters. Dopamine is a major one here.
Dopamine, the reward chemical.
Exactly. Associated with pleasure, motivation, that wanting aspect of desire, it's thought that PT-141 influences dopamine release in specific spots, like the medial preoptic area or MPOA. The MPOA is a known hub in the hypothalamus for regulating sexual behavior and motivation.
Okay, so activating MC4R leads to changes in dopamine in these motivation centers.
That seems to be the leading hypothesis. And by doing that, it's believed to sort of enhance the salience of sexual cues. It sharpens attention towards them and, really importantly, amplifies the overall motivation, the drive for sexual activity.
So it's not just a general feel-good effect, it's more directed.
That's the idea. It seems more targeted towards that appetitive drive. Both animal and human studies point towards MC4R signaling being this crucial link between what's happening centrally in the brain and the actual subjective feeling of desire.
That helps clarify a lot. So going back to our analogy, if the vascular approaches are like opening local bites, how would you describe PT-141's action? Is it really just flipping a switch?
Well, maybe flipping a switch is a bit simplistic, but it captures the central idea. A better analogy might be, if vascular approaches are local plumbing, PT-141 is more like a conductor leading an orchestra.
Okay, I like that.
It's coordinating different sections, various brain regions, neurotransmitter systems, to create this focused outcome. Desire. It nudges the brain's own control centers, fine-tuning that motivational system specifically towards sexual interest and drive.
A conductor, not just a plumber. Got it. That makes the difference really clear.
It's fundamentally different, yes. Brain-driven arousal.
Okay, this is fascinating. Let's talk about the evidence then. What do the actual studies show? Especially, you mentioned trials in women with HSDD.
Yes, the research in women, particularly those Reconnect studies, provides some really compelling data. These were two large, well-designed trials, Phase 3, randomized, double-blind, placebo-controlled, the gold standard, really.
Right.
They focused on premenopausal women diagnosed with hypoactive sexual desire disorder, HSDD. And HSDD, just to clarify for listeners, isn't just low libido. It's a persistent lack of sexual thoughts or desire that causes significant personal distress.
That distress component is key, isn't it?
Absolutely. It really impacts relationships, self-esteem, quality of life, and treatment options have been, well, limited.
So what did the Reconnect studies find with PT-141?
They found statistically significant improvements compared to placebo. This was measured using validated questionnaires, things like the Female Sexual Function Index, the FSFI.
Okay.
Specifically, the desire domain score on the FSFI went up significantly for the women using PT-141. And crucially, they also saw a significant decrease in the distress associated with low desire, measured by the Female Sexual Distress Scale, FSDS.
So improvement in desire and reduction in the distress it caused.
Exactly. Both endpoints were met. And these improvements were seen over the 24-week study period, suggesting a sustained effect. For women struggling with HSDD, this was quite significant news. It pointed towards a potential treatment targeting those central brain pathways, offering a different approach.
That's powerful, especially linking it to reduced distress. Now, you mentioned it was studied in men, too, in early trials.
Yes, there were earlier controlled studies in men as well. They explored different delivery methods, intranasal spray, and subcutaneous injection under the skin.
And what did they find there?
They observed dose-dependent erectile responses, meaning higher doses tended to produce a stronger response compared to placebo.
Okay, so a direct physiological effect.
Yes. And what was quite striking was the onset time. Effects were sometimes seen within tens of minutes.
Wow, that fast. For something working in the brain.
Relatively fast, yes, which suggests a pretty direct engagement with those neural pathways controlling arousal. And importantly, these responses were often measured objectively, using devices like Rigiscan that track penile rigidity and duration.
So not just subjective reports.
Right, quantifiable data. This was important because it helped differentiate this central melanocortin approach from purely peripheral ones like PDE5 inhibitors, which mainly work on blood flow locally. It showed you could potentially trigger a physical response by influencing the brain's control centers first.
That really reinforces the central mechanism idea, even for a physical response like erection.
Precisely. It highlighted that central pathways could be key, even when maybe peripheral treatments alone aren't enough.
Which leads perfectly to my next question. If it's working so directly in the brain, can we actually see it happening, like with brain scans?
Yes, and that's where things get really visually compelling, you could say. There was a key fMRI study, functional magnetic resonance imaging. It essentially tracks changes in blood flow in the brain, showing which areas become more active.
Okay, so they put people in an fMRI scanner after giving them PT-141.
Exactly. It was a double-blind, placebo-controlled crossover study, so quite rigorous. Participants got PT-141 on one occasion, placebo on another, and their brain activity was scanned. And what they found was fascinating. After PT-141, there were noticeable changes in brain activity, specifically in regions known to be involved in processing sexual cues, self-awareness, emotional processing, areas like the insula, anterior cingulate cortex.
The kinds of areas you'd expect to be involved in desire and arousal.
Exactly. And crucially, these changes in brain activity correlated directly with participants reporting increased sexual desire.
So the brain scans matched what people were actually feeling.
Yes. A direct link between the neural changes and the subjective experience. And these effects were observed for up to about 24 hours after just a single dose.
24 hours, that's quite a duration from one administration.
It suggests a significant period of neural modulation, yes. So this fMRI data is really valuable. It gives us objective, brain-level evidence that aligns with the subjective reports and the proposed mechanism. For you listening, this shows it's not just a mood thing. It seems to be actively changing how these specific desire-related networks in the brain are functioning.
That's incredibly powerful, seeing the brain light up, essentially. Okay, and I think you mentioned some research is looking at combinations now. What's the thinking there?
That's right. The field is evolving, and researchers are exploring if combining approaches might be beneficial. One current area is looking at co-administering PT-141 with PDE5 inhibitors.
The drugs typically used for erectile dysfunction.
Correct. Those work peripherally on blood flow. The idea is being tested, particularly in men who don't respond well to PDE5 inhibitors alone. These are often considered tough cases.
Why might someone not respond to those?
Various reasons. Could be severe underlying vascular issues, nerve damage, or sometimes the central drive, the desire component, is low, so the peripheral drug doesn't have the signal to work with, if you like.
Ah, makes sense.
So the rationale for combination therapy is, what if we engage both the central pathway with PT-141 to boost that desire signal from the brain and the peripheral pathway with a PDE5 inhibitor to support the vascular response?
Covering both bases, essentially.
Exactly. The hypothesis is that this dual approach might provide a more robust or consistent outcome, especially for those non-responders. Now, it's important to stress this is still very much in the research phase, investigational.
Got it.
But it shows how the thinking is evolving, trying to tackle the complexity of sexual function from multiple angles, recognizing both the brain and body components are crucial.
That makes a lot of sense, especially for those difficult cases. Okay, so we've covered a lot, the unique origin, the central mechanism, the different strands of research in men and women, the brain imaging. If you were to synthesize all of this, pull it together. What's the big takeaway? What do these findings collectively suggest about PT-141?
Well, pulling it all together, what consistently emerges is PT-141 acting as a central pathway modulator via that MC4 receptor. All the evidence points in that direction. The improvements in desire and distress scores in the women's HSDD trials, the fMRI data showing activation in those specific brain regions linked to sexual processing and desire, and even the rapid dose-dependent responses in the early male studies. They all align with the central brain-driven mechanism. It suggests PT-141 is a really interesting tool for understanding how our brain signaling directly influences arousal. It moves beyond just blood flow. And the fact that research is now exploring combinations, especially for hard-to-treat groups, shows there's ongoing interest in leveraging this central pathway, perhaps alongside other approaches. So the bigger picture is, it really underscores the importance of the brain in sexual function and desire, and PT-141 continues to be a key focus for research in exploring that connection.
That's a great synthesis. It really highlights that brain-first perspective.
Yeah.
So as a neuroscientist working in this area, what excites you the most about PT-141 and what it reveals? Thinking bigger picture.
For me, it's exactly that bigger picture. It's less about the peptide itself, maybe, and more about what it tells us. It really drives home the point that sexual function, desire, it's not just plumbing, it's not just mechanics. It's deeply interwoven with our brain chemistry, our motivation systems, our reward pathways. It's a complex neurological event. Yeah. And by studying things like PT-141, we get this window into how the brain and body talk to each other, how they integrate to create these experiences. It highlights the sheer complexity, but also the elegance of human biology. And ultimately, this deeper understanding, it could really shape future approaches, potentially leading to more targeted, more effective ways to help people by addressing the central drivers, not just the downstream symptoms. That potential to improve understanding and maybe treatment based on this deeper neurobiological insight, that's what's really exciting. What stands out most to you? Haven't heard all this.
That's a great question to reflect on. I think for me, it's that bridge between the subjective and the objective you mentioned earlier. Seeing something as personal and, well, intangible as desire show up as concrete changes on a brain scan. That's just mind-blowing, really.
It is quite remarkable.
It makes it feel less like a mystery and more like a biological process we can actually understand and potentially influence. That connection is what really stands out. Well, thank you so much for walking us through all of this today. Your insights have been incredibly valuable, making this complex topic so much clearer and, honestly, really engaging.
It was my pleasure. It's a fascinating area.
And to all of you listening, what a deep dive that was. The key takeaway. PT-141 is being studied for its unique role in the brain's central pathways, engaging motivation and reward systems directly, a very different approach than just focusing on circulation. Hopefully, this has given you that shortcut to being well-informed on what is truly a groundbreaking area of research. Now, just a crucial reminder for everyone. The mentioned products and peptides are not intended to refer to any peptides or products sold by Power Peptides. Products sold are strictly for research purposes and not intended for human consumption. If you're looking into research-only materials, please check out their site responsibly. Okay, so here's a final thought to leave you with as you go about your day. If desire truly is, as our expert suggested, a product of our brain's complex orchestra, how might understanding these central conductors better? How might that reshape our view, not just of sexual function, but maybe of human connection, motivation, the whole experience itself? Something to ponder. Thank you for tuning into The Deep Dive. Make sure you subscribe so you don't miss our next exploration. Until then, stay curious.